that is molecular basis of inheritance. In the previous video, we have seen several topics, and if you wish to check all those topics, you can check my description box for it. Today we are going to see the next topic of this chapter, that is properties of DNA, right? Now we have seen the structure of polynucleotide chain, we have seen several experiments, right? We have seen how the DNA is packaged, right? Today we are going to see that what exactly the differences between the RNA and the DNA, and what are the main properties of DNA and how it is more stronger and more stable than RNA, right? So the first, it should be able to replicate, right? Now the word replicate means to duplicate itself, right? That means to produce same exact copies of the DNA, right? So a DNA should have a property of to replicate, right? That is, it should have ability to replicate, that is duplicate. It should be chemically and structurally stable, right? Now the DNA should be chemically, that means the chemical composition of the DNA as well as the structure of the DNA, both should be stable. If there will be no stability, then the particular DNA cannot work or it cannot produce its exact identical copies, right? So for this reason, they should be chemically as well as structurally stable. It should have scope for changes, right? Now the changes here, the word changes determines that mutation, right? That means for the evolution some changes are needed, right? For example, if the humans in the previous era, right, they were comparatively so different than the humans today are, right? That means several types of changes are taking place, and those changes are nothing but it is carried out by a mutation, right? So it should have, the DNA should have the ability to mutate itself. That is the changes which is carried out that are essential for evolution, right? For carrying out or for occurrence of the evolution, the changes or the mutation should take place, and that should be done by the DNA, right? It should follow Mendelian principles of inheritance, and the last characteristic of the DNA is it should follow Mendelian principles of inheritance. That means it should have the ability to pass the genetic information from one generation to the next generation. So these are the main characteristics or properties of DNA, right? Now we are going to see the difference between the DNA and RNA, right? The DNA has deoxyribose sugar. The full form of DNA is deoxyribonucleic acid. And when we are talking about RNA, the full form of RNA is ribonucleic acid. So the DNA, as it is deoxy, right? That means it has ribose sugar, but it is a deoxyribose sugar, right? When we are seeing for the RNA, it is containing what? A ribose sugar. So the first difference between the DNA and RNA is the DNA contains deoxyribose sugar and the RNA contains the ribose sugar. Then DNA has thymine as nucleotide, while RNA has uracil in place of thymine, right? This difference we have seen previously in the introductory part when we have seen the structure of polynucleotide chain. That the DNA contains thymine, the 4 nucleotides is ATGC. That means the DNA has different nucleotides and those are ATGC, that right? And when we are seeing about the RNA, it also contains AUGC, right? That is, instead at the place of thymine, what is present? Uracil. So DNA contains the thymine, and in RNA, what has present? What it is present? Uracil, right? The third difference, that the DNA mostly acts as genetic material. Now, normally the DNA is mostly acting as a genetic material in most of the organisms, right? But when we are talking about RNA, it is present in few viruses as a genetic material, but mostly they act as messenger and adapters, right? That means they are carrying out the passing of the messages, right? That is messenger. It is a type of messenger which is transmitting messages from one to the next, right? So they mostly act as messengers as well as adapters to bind them, right? The DNA is stable, right? And when we are talking about RNA, it is level labile and biodegradable, right? When we are talking about DNA, it is stable because it does contain two strands, right? And they are intertwined with each other to form a coiled structure. That means they are easily not degradable, right? Or we can say they cannot easily unwind itself. For this reason, they are more stable compared to RNA. The RNA is labile and biodegradable. Labile, that means it is a type, or we can say it is unstable, right? Why it is unstable and biodegradable? Because it contains a 2' OH group at all the nucleotides, right? And for this reason, small things are being produced, right? And for this reason, it is said that it is biodegradable, right? In DNA, what happens? The genetic information is stored, right? So the function of DNA is to store the genetic information. And what is the function of RNA? The transmission of genetic information. So the DNA's function is to store the genetic information, and the function of RNA is to transmit the genetic information, right? Then DNA is chemically less reactive and mutates slowly, right? What happens in the DNA? It is chemically less reactive. That means the reaction which is taking place when we are seeing the chemical composition at that time, chemically they are less reactive, and for this reason then the mutation which is taking place, it is also comparatively less, right? And when we are talking about RNA, they are chemically reactive and they mutate fastly, right? The DNA, when we saw DNA, the mutation takes place slowly, and in RNA the mutation takes place fastly. And as DNA was less reactive, here RNAs are comparatively more reactive, right? The last difference of DNA is that DNA requires RNA for protein synthesis, right? The central dogma says that the DNA needs a RNA to produce a protein, right? That means the DNA converts to RNA, the RNA converts to protein. So for the formation of the protein, this DNA needs what? A RNA. But when we are talking about RNA, the RNA directly codes for the protein synthesis. So this is the main difference between them, that the DNA needs RNA for protein synthesis, while the RNA can itself produce a protein, right? So these are a few points of the difference between the DNA and RNA. Now we are going to see a most important question, that is why DNA is more stable than RNA, right? Now we have seen already the difference. You might have several things in your mind that we have already seen the difference, and amongst this difference we can carry out or we can understand that why DNA is stable. But we are going to see few steps over here, right? So in this few points we are going to see that how the DNA is comparatively more stable than the RNA. So in RNA, a 2' OH group is present at each nucleotide. Now at each nucleotide, at every nucleotide, that is A, U, G, C, the RNA contains what? A 2' OH group, right? That is 2' OH group. And due to this, what happens? It makes the RNA unstable and degradable. That means the easily degradation can take place, and it is comparatively unstable than the DNA. The presence of thymine in place of uracil confers additional stability to DNA. Now the DNA is comparatively more stable because the presence of thymine is there, right? The uracil is present in RNA and the thymine is present in DNA. But when the thymine is present in DNA, it can easily bind with the adenine and it forms a helix structure, and it provides additional stability to the DNA. RNA being a biocatalyst is more reactive, right? Now as the RNA is said to be biocatalyst, biocatalyst means it can carry out the reactions very fastly, right? It is a kind biocatalyst enzymes, right? You might have heard about enzymes, and enzymes are biocatalysts, and the enzymes are responsible for the increasing or for producing more amount of reaction, or we can say it increases the rate of reaction, right? So RNA is a kind of biocatalyst, and for this reason they are comparatively more reactive. DNA is double-stranded, having complementary strand which resists the changes by the repair mechanisms. As the DNA contains two strands, for this reason when the mutation takes place, what happens? It can be easily repaired by the repair mechanisms, right? And for this reason, as the RNA is not containing another strand, and for this reason there is no chances of repair mechanisms in it. And when we are comparing that with the DNA, it does contain two strands, and for this reason the repair mechanisms can be easily done. And this way the amount of mutation which is taking place can be controlled, right? So this is all about properties of DNA, and which is a genetic material, right?